Good afternoon. We are going to start the second part of our symposium on neotropical subtrial line. It is a pleasure to introduce Professor Cássio van den Berg from the Universidade Federale de Feira de Santana, Bahia, Brazil. Forget my Portuñol. Welcome, thank you.
Okay. Well, good afternoon, everybody, and thank you for being here. And I would like to start thanking the committee for inviting this talk and Gerardo who organized the symposium. And today I'm speaking about Cattleya, and Cattleya phylogeny is something very interesting. I didn't speak about it since my PhD 25 years ago. We did a few things on Cattleya, but actually I stopped doing things with Cattleya and we returned to do this in the pandemic, during the pandemic. So this is pandemic science, really. And we started to do things again and to check on the phylogeny of these groups. Well, the Cattleya, I'm not talking only about Cattleya. The title of the talk was wrong, but it's actually the whole of the Cattleya alliance, which are a few genera. This is the only published Lelini phylogeny. You can see that actually is quite unresolved as for the spine of the tree, but we had actually several alliances which were supported in this paper. And actually you see that the Cattleya alliance is actually in a zone of the tree. You have more than 90 or 95 of posterior probability. This is a Bayesian tree. And one of these groups is the Cattleya alliance. And the Cattleya alliance is basically 3 genera, which are Rhyncholaelia, Guarianthe, and Brassavola, and all the rest are Cattleyas. And when we did the Sanger sequence in these phylogenies, we actually put all these things in red here, are actually plants that belonged to the genus Lelia and were transferred to Cattleya. These are all Brazilian groups of species. And of course, the true Lelias are in Mexico, and there is nothing in between. So they had been for a long time starkly kept in Lelia by morphological characters, but when we did the phylogeny, they actually have nothing to do with Lelia. They are not even in the Lelia alliance of Lelini. And one thing that you see quickly in this tree, that there is one subgenus here, which is Maxima, has a single species, and it goes here with the plastid. But if you include ITS, it has actually ITS of the very last group of the phylogeny. So it is on the opposite side of this phylogeny. So there is a very strange conflict here. So that's one of the questions you want to address with more data. This is the diversity of Cattleya. So in 2014, despite that conflict, we divided the groups in a way that was compatible with both systems. And we established 4 subgenera. So we have one here that's about 25 species, 2 that are monotypic, and then it has this huge subgenus Cattleya, which in turn, it is divided in 3 sections. So you have one section with these species here, a small one here, and you have this whole section, which is equivalent to the old Brazilian Lelias and Sophronitis. So the 2 genera that were moved in Cattleya go into a single section. And so this section that came from Lelia and Sophronitis is divided in 5 series. So we actually established a formal taxonomy based on on the old phylogenetic results we had. So why, if we have this, all seems quite, the group seemed quite homogeneous, but why so we need to do phylogenomics of the Cattleya alliance? Well, actually, we always had some problems knowing what's the exact relation between Cattleya, Brassavola, Rhyncholaelia, and Guarianthe. And Guarianthe is a genus that we never managed to make the 4 species come together with Sanger sequencing. There was always a polytomy. So that's one of the questions. And of course, we would like to have a very well-supported relationship of all these infrageneric taxa within Cattleya. So we really want to get well-supported relationships with all the subgenera, sections, and series. But the main reason really is that I'm showing you all these phylogenies of the larger groups, but we never managed really to get very well-resolved species phylogeny within any of these subgroups. And so that was actually the third one, was the main motivation. If I show you, for example, this bifoliate Cattleyas, which is one of the subgenus from Brazil, and I had a student in 2011 that he did 14 plastome regions. There was MATK and 13 intergenic spacers and non-coding DNA. And this group starts about here. So you had some small groups that were resolved, but most of it is a polytomy. And it's the same pattern you get with nuclear data. So if you take ITS, and we managed to have one nuclear gene at that time sequenced, and more or less the same group gets resolved here, but you have big polytomy on the top. But that's actually a good one, because and one thing that I want to show you as well, that there is some conflict as well between nuclear and plastome data. So you get actually, the right side here is actually the bifoliate ones. So some species being suggested to be hybrids from old hybrid origin based only on conflict between plastid and nuclear genes, but there wasn't really good support to do much with this tree. So we don't believe it very much because the data was not very, that didn't have a lot of resolution anyway. This is the outgroup part, so you just forget about it because this is already, it's very badly sampled, so we cannot really rely on anything on this side of the tree. Well, in 2019, I had another student that decided to, we decided to do this Rhyncholaelia Cattleya. This is a small group that grows in rocky outcrops in Brazil, and is the most diverse lineage of Cattleya. It has about 60 species, and that in several different color groups, and they all grow on rocks, and most of the species grow in islands, and they are endemic to a single mountain and things of that type of thing. And she did 6 plastid spacers. And you see this tree, and actually if you look at the support here, even the part that is resolved, it's not resolved at all. It's like 50, 0.5, 0.6 for Bayesian analysis. You could consider this whole tree is more like an unresolved tree, and maybe the only things that are resolved are when you have 2 samples of the same species. So this student was in despair, and she finished her PhD thesis doing a morphometric work, because so she did like morphometric work for species delimitation, because you couldn't really publish this thing because it's just like a general comp. So, but when I say that, so let's say this group specifically was the one that got me motivated to go after phylogenomics, because when the pandemic started, I decided to move closer to my parents because I lived very far away, and I got a visiting position in the University of São Paulo that was close to my parents. And then I got a lot of time to go to the wild and recollect all these samples and get a much bigger sampling. And then I got also 2 students in São Paulo that were interested in doing this work, and then we started to do population genetic works in several of these groups to do species delimitation. And then we had a lot of extra space on the NGS plates that we could do some extra work, and I decided to do phylogeny with these extra spaces we had for doing population genetics. So this very group here is the one that we started us into doing phylogenomics of Cattleya, but then we ended up sequencing all the species of Cattleya and all the species of the outgroup, so we had no species missing in the phylogeny of the whole of the Cattleya alliance sequenced in the end. So what we did here I put first low coverage sequencing, but we actually started with ddRAD because we were doing ddRADs to do the population genetics. But we ended up doing phylogeny with the ddRAD as well because it was very promising. But we also had some extra space in the plate, so we did some low coverage sequencing as well. So I'll show both results here. And I like to show people, sometimes they ask me how much this costs. So actually doing the low coverage is a bit expensive, it's like nearly $200 per sample. But doing the ddRAD in our lab and doing ourselves the libraries, the cost of the RADSec was about $12 per sample. So it's the same cost we spent to sequence one sequence of ITS. So it's extremely cheap way of doing NGS if you do it yourself. If you hire a company to do it, they charge you about $50 per sample. And well, so I start to show you quickly the results of the low coverage sequencing. In the end, we managed to do 27 plastomes, and they are about 157,000 in size. Well, this is the line size, so they're about 153 each. So this is because there's some extra gaps, so they get to 155. And they provide about 2,300 variable characters, well, like parsimony informative characters. So that's not only variable, but the ones that you can use to infer phylogenies here, really. If you take only the coding genes, you get actually only half of it. So I'll show you both trees. You see that you really need to use the full aligned plastomes because a lot of the information is really in the non-coding part if you're dealing with closely related species. And we also, with the same data, we assembled the complete nuclear ribosomal DNA that gives a matrix nearly 11,000, and it's got even slightly more characters than the coding genes of the plastome. Okay? So here I'm showing you the two trees. Let's say if you do the whole of the aligned plastids, and if you do only the coding genes. And with the aligned plastids, actually, you get a lot of support here. And you see I marked in green, but the ones that are not green are still 82, 97, 99. So there's really only one relationship that is a bit undefined here. But you get two groups of Cattleya here, right? And of course, all the old Lelias and Sophronices cluster together. And you get more or less the same relationship here, but you also get the same problem of support here, but then you get a lot of low support there if you discard the non-coding parts of the plastids. And one thing that is strange, let's say, you start to get strange relationships. For example, Guarianthe, if you discard this data, it goes very near Cattleya here, and Brassavola goes here, but really if you have all the data, you have Brassavola as the outgroup of outside everything, then Guarianthe, then Cattleya. Unfortunately, the sample of Rhyncholaelia failed here, so we didn't put another one yet. If you do the nuclear ribosomal DNA, I had a lot more taxon in Lelini here, and actually Myrmecophila didn't go with Berkeria with this data. It actually went closer to the Cattleyas, but it didn't go with much support here. So it's actually more like a non-resolved relationship comparing to the talk we saw this morning, right? You don't get all that many supported clades, but you still have a lot of support for the infrageneric groups in the same ways as in the plastids, okay? So I'll show you a bit now what the results we ddRAD are. And ideally, when you do RAD sequencing, you would like to map the reads to a reference genome, but there's no reference genome for Cattleya. So we have to do de novo assembly. And the thing with de novo assembly, you need to do quite a lot of tedious bioinformatics with this. Because basically the way of doing RADSec, you digest your, people who are not familiar, you digest your DNA with two restriction enzymes, and then you ligate adapters. and basically amplify this with PCR and sequence in Illumina, right? In our case, we have all the fragments are single end, and they have 100 base pairs. And then what you have to do a clustering of these reads. So let's say if I choose a clustering threshold of 90%, I will consider that any two reads that have less than 10 differences will be a single locus. So that's what we do. And basically, you have to cluster it first within species and then between the species. And in this case, you have actually to optimize these things twice because we have two clusterings. And this is a very important thing to take care, because if you cluster too low, if you cluster very different things, we end up putting two paralogs as a single locus. And if you cluster too high, then the same locus will become like two loci, and then half of the taxa will be missing data. So you always have to be finding an optimal position with this. Well, the nice thing with this ddRAD is actually that you can reanalyze your data several times from the reads. So if I take all the 240 taxa, I get about 98,000 sequence matrix. But if I restrict to a subset of samples, then I got like 1,017,1,600 and things. So you can get a very high number of characters by getting closer, because you have a lot more restriction sites which are shared between your taxon. So this is the main tree with all the 140 terminals that I will resume here. So the same tree, I just collapsed the things. And you basically get the same phylogeny you would get with the plastid. So it's actually very, very nice. And Brassavola is really recovered as a sister group. And I just want to show you something. One of the problems of doing this is, although you get like twice as much data as the plastids, if you want to go for coalescent analysis, you have to take only the unlinked loci. And then that's actually a big limitation of RAD-seq, because you cannot do astral with it, because the loci have too little variation. So it's a bit of a, and if you get only the unlinked loci, you get much less data, and you actually don't get all that much support. So it wasn't very convincing to do the coalescent analysis here. Well, I'll show now very quickly to species phylogenies just in these subgroups. Then you get a lot of data, and basically you get very well resolved species phylogenies, because you get nearly 100% in all in nearly all the nodes. This one I chose to show you just because it includes the Guarianthe Skinner, which is the national flower here. So you see that actually Rhyncholaelia is confirmed here as sister to Guarianthe. These are outgroups, and you have a phylogeny of Brassavola, for example. And one of the tricky things I show you is this Cattleya raguaiensis. This species is a bit like that hybrid of rodent with Berg that Clarice showed in the morning. It's a very strange plant. And it has these fibers with ellipsoid silica bodies in the leaves that are found only in Brassavola, despite being a Cattleya. So we always suspected that this could be a hybrid of Brassavola and Cattleya that goes basal. And then you can do actually a PCA of the SNPs, and the data really doesn't confirm that, but it doesn't have any sign of Brassavola genome. So see, it goes very closely in the axis that separate Cattleyas from Brassavolas. It goes together, so it's probably just some sort of ancestral character. Another example here I'm showing you is the large Cattleyas, the Cattleya section Cattleya, and you get a lot of support in most of the relationships, and they actually make an arch. So you get plants from Peru, from Western Colombia, then Central and Eastern Colombia, then Venezuela, then Brazil. So this is a very nice group to take this data and do now biogeography. You just need to recalibrate these trees with secondary calibration from the plastid phylogeny that you can take up to the fossils. Here I'm just showing you another 3 examples. So you get extremely well-supported phylogenies for all the species. These are all the species that exist in the whole of the Cattleya alliance. They are in these 5 trees, the previous slides in these 3. And that's actually, you get a lot of well-supported data, maybe not so much in that Rhyncholaelia that started the work. So you see that actually they have maybe too many species for the radiation we are dealing here. So this group may still be a problem, but at least it is divided now in 3 main clades of species, right? So just to show you that this is very promising, we have to have students to do 1 paper with each of these phylogenies because the biological questions in them are completely different. So just make some final conclusions here. ddRAD will be very inexpensive. So today it's not worth doing an ITS. It's better to do a ddRAD instead because you have a lot of characters and spending the same price. But you have to have this kind of DNA. So if you have bad DNAs, you have to go to Bates. There's no way because you need intact DNAs to do the restriction sites to do this. The bioinformatics is a bit tedious, but now we are doing a reference genome to make it slightly better because they say that actually improves even more these results if you map it to a reference genome. This is actually a limitation. This limitation to use SVD quartets means that you have to have a lot of SNPs to select these unlinked SNPs to be able to do a decent coalescent analysis. So this is actually a problem. But if you have to do a lot of samples or a lot of species, and you're doing species delimitation either by population genetics or by just making multiple samples of each species, that's probably a very good and cheap way to go. And one thing is that because of the closest your plants are, the more restriction sites and loci will have. So that's one interesting thing, that this method, the worst things go with the other methods because they lack variation in any other methods. The best they will do here. So it's actually a very interesting approach for people who are interested in doing it. So just finish here thinking. I have 2 students involved in this project, and Maria is a researcher who received me in her lab in São Paulo when we were doing this work. And I had 2 orchid amateurs who provided a lot of samples, and they will be co-authors in the paper because they were very important for the work. Thank you.
Now we have time for 1 question.
Thank you very much. It was amazing. I would like to know what's the enzyme combination you use for ddRAD?
Oh, I put it there. It's PST1 with MSE1. This combination, because if you change the combination, you have to do or redo all your adapters, and that's quite expensive because we are doing like 384 plates. Yeah. So you have to make 384 adapters and anneal them all and requantify. It's actually very laborious as well. We noticed in the lab I was first doing this, they were working with palms and with several other groups of plants, of insects, and all the things that are about genomes about 1.5 to 4.5 gigabytes do very well with this enzyme. Actually, one of the plates, I had another study with rattlesnakes. So we put half of the plates with rattlesnakes, which have a 3 GB genome like human genome, and they actually provided a huge amount of SNP data. And we are doing the same combination in palms, and it works wonderfully well. So we basically, when we found this combination of enzymes, we noticed that if we are dealing with Lelini, for example, they have quite similar genomes in size, not in Barcaria. But we can keep doing a lot of study using the same pair of enzymes.
Thank you very much.
Thank you very much, Cassio.